So the recording is in process and you can start from here, sir. Over to you. Sir, you can start with the introduction and all. You need to unmute, sir.
Okay.
Please proceed.
Good evening and warm welcome to you all. I am Dr. Joel Stewart, recent year student from Tagore Medical College and Hospital, Chennai. I am really elated to see all the medical students and doctors across the length and breadth of this nation, those who joined in this webinar today. IMA-JDN has been doing a plenty of webinars on various topics with the objective of disseminating knowledge and keeping the medical community informed and updated. IMA-JDN, in collaboration with SNO Tamilnadu, under the guidance of Dr. Abul Hassan sir, presents a webinar on the topic spinal cord and its internal features. The patrons of our webinar are Dr. Abul Hassan sir, a well-known pediatric surgeon from Erode. Sir is the chairman of IMA-JDN Tamil Nadu and also the IMA-TN State President-elect 2024. I would also like to mention the organizer of today's session, Dr. Venkatesh Karthikeyan sir, a community medicine and family medicine PG from AIMS, Patna. I feel honored to introduce the speakers of today's session, Dr. Surya Karthikeyan ma'am, MD Anatomy. Now she is the assistant professor of Government Ooty Medical College, Delhi, Greece. I request Dr. Surya Karthikeyan ma'am to take over the session. If you have any queries, kindly leave in the chat box. The queries will be answered at the end of the session. Ma'am, over to you, ma'am.
Thank you, Dr. Joel. Yeah, shall I start the session, Devlin?
Yes, ma'am, you can proceed now.
Okay.
Yeah. Is it visible? The screen is visible now?
Yes, ma'am. The screen is visible.
Yeah. Okay. Good evening to everyone. Welcome back. So I'm taking a series of sessions on neuroanatomy. And a small correction, the last class was spinal cord and its internal features. And then today's class is about cerebral cortex, mainly about the functional neuroanatomy. And then I'm going to start my session now. So again, anatomy and then medicine, it's actually a boring topic. And then it's very complex to know all those things, right? That too, neuroanatomy. Whenever I start the class, I'll tell them it's very complex to understand all these structures, to memorize all these structures, to remember all these things. But when you understand this, you can actually, it's easy to memorize also. It's easy to remember all those names also. So I'll start the cerebral cortex. So today's class, at the end of this session, you will be knowing a little bit of embryology. I'm not going to talk in detail about the embryology, but it is about little bit embryology and then sectional anatomy. Because whenever we are reading the cerebral cortex or whenever we are reading about the brain and all neuroanatomy, they will explain the things in different sections. Like it can be a cross section, it can be a horizontal section, or it can be a vertical section, that is sagittal, and then you have coronal sections also. So we need to know about all the sectional orientation. That is the basic anatomy we need to know. When with that knowledge, we can actually apply that knowledge in the radiology. Okay, that is, you can apply that in CT, MRI pictures and all. So you need to know about the sectional anatomy. And then I'll be talking about gross anatomy and then some evolutionary aspects. That is, over a period of time, our brain has evolved. Okay, it has undergone so many changes. So we'll be seeing about a little bit changes about evolutionary aspects. And then I'll be talking about lobes and then white matter of cerebral cortex. Also, I'll be talking about the functional areas, maybe the functional areas of cerebral cortex. And then finally, you can actually have a review on clinical anatomy and lesions of cortex. Okay, so the session is going to start now. A little bit numerical data. What is the total surface area of cerebral cortex? What about central nervous system? The central nervous system, it constitutes both the brain and the spinal cord. Okay, in the brain, I am talking only about the cerebral cortex. This class, I'm going to talk only about the cerebral cortex, but we have other parts of the brain, that is cerebellum, brain stem, and then we have in the brain stem, we have the pons, medulla, and the midbrain and all. But only I'm concentrating about the cerebral cortex today. Okay, what is the total surface area? It is around 2,200 centimeters square. What we are seeing, the surface area, is around only about 1/3 of the cerebral cortex, but about 2/3 is hidden in the sulci. About 2/3 is hidden in the sulci. Okay, the thickness of the cortex is around 1.5 millimeter and it weighs around 600 grams. That is, the cerebral cortex takes up at least 40% of total brain weight. When we weight the total brain, only the cerebral cortex is taking about 40% of its weight. That is around 600 grams, in which only 180 grams is occupied by the neurons. We'll be thinking, it's very fascinating, no? Actually, neurons are the functional unit of brain. We all know it is a functional unit of brain. So we will be thinking that the cerebral cortex is actually consisting of so many neurons, nerve tracts, fibers and all. But only 180 grams is occupied by the neurons. It is supported by the supporting cells. That is the neuroglial cells, which takes up around 420 grams of the weight. Okay, 180 grams is neurons and 420 grams is occupied by the glial cells. That is the supporting cells. Okay, so this is a beautiful picture. This image we are seeing from the posterior view. Okay, from the posterior view, we are seeing this image. Here they have made an incision along the midline. Posteriorly, they have made an incision along the midline and they have decoded all the cranial cavity, all the meningeal coverings and all. So when I was talking about the spinal cord itself, I told you we have the meningeal coverings for the brain and the spinal cord. So what are the meningeal coverings? You know, we all know it has an inner pia mater and then arachnoid matter and then outer dura mater. There are three meningeal coverings: pia mater, arachnoid matter, and the dura mater. So they have reflected all the pia mater, meningeal coverings, and then they have shown the brain here. Okay, this is the cerebral cortex and then cerebellum, and then it is continued as spinal cord. So from the last class, you all know the spinal cord ends at the level of lower border of L1 in adult. But whereas in newborn, it is at the level of L3. Okay. And then, but the meningeal covering extends till the S2. And then we have the modification of pia mater. That is the phylum terminal externum. It is extending till the coccyx also. Okay. We have the spinal cord till L1 level, meningeal covering till the S2 level. But the modification of pia mater, that is phylum terminal externum, it extends till the coccyx. Okay. Okay. This is the same picture from last class, that is about embryology. If you would have remembered, this is very better, but I'll revise again. Okay, so what is this position? So this is the developing embryo at different stages. The developing embryo at different stages. So what is this view? We are viewing the embryo from the superior aspect. That is, we have removed the amniotic cavity. We have removed the amniotic cavity and then we are viewing the ectodermal part. Okay, the embryo, it is actually covered by an amniotic cavity and also it has an yolk sac in front. So what it is imaged here is they have removed the amniotic cavity and what we are seeing is the craniocaudal orientation of an embryo. So this is cranial end of embryo and this is caudal end of the embryo. Okay, so what happens is during the development, there is a thickening in the ectoderm. Okay, there is a thickening in the neuroectoderm that is called as neural plate. The thickening in the neuroectoderm in the midline, it is called as neural plate initially. And then in next stage, what happens? It depresses. It depresses to form a groove. Okay, so this is a cut section. This is a cut section of the embryo where you can see the groove. Okay, the neural plate has deepened to form the groove. So that what happens is on either side it has formed a fold. Initially it is only a depression, it is a plate-like structure. But when it deepens, what happens is on either side it is going to form the folds. So those are called as neural fold. So in next stage what happens? The neural fold, it unites to form a neural tube. So when you take a paper and then in the midline, if you depress both the ends, it will be forming the neural fold. So when you bend the paper posteriorly, what happens? It will be forming a tubular structure. The same way, the neural plate depends to form the neural groove, and then the neural groove, now it has formed the neural fold. The neural fold united to form a tubular structure. Okay, so this tubular structure, it is open at both the ends. Along the cranial end also it is open, and then caudal end also it is open. And then in next stage, in further stages, what happens? The neural tube completely closes, and then the cranial neuropore also closes. At what day of intrauterine life the cranial neuropore closes? This can be asked as a neat question for you. So the cranial neuropore closes at around 25th day of intrauterine life. And then caudally, the neuropore closes at around 28th day of intrauterine life. Okay, this is caudal neuropore, which closes around 28th day. This is cranial neuropore, which is closes around 25th day of intrauterine life. Okay, so in last class also I told you the cranial end, now it is going to form the brain. Whereas the caudal part of the neural tube, it will elongate to form the spinal cord. Okay, so the somite formation also it goes on. Simultaneously, the somite also it is formed. Okay, the somites will be going to form the vertebra under. So inside the vertebral column, the spinal cord will be formed from the caudal part of the neural tube. The cranial part is going to form the brain. So what happens in next stage? This is about developmental anatomy. What you are seeing is the embryo at 4 weeks of gestation. So embryo, they are imaged. These are all just schematic diagrams to understand. Okay, this is the cranial part of the neural tube where it is distinguished into forebrain, midbrain, and then hindbrain. In next stage, the cranial end as distinguished into forebrain, midbrain, and then hindbrain. Now we have 3 parts. Okay, what happens in next stages? It is going to form another 5 vesicles. These forebrain, it will be subdivided into 2 more things. Okay, that is telencephalon and diencephalon. Midbrain, it will form the mesencephalon, whereas hindbrain, it is divided into metencephalon and then myelencephalon. So from cranial to caudal, the 5 vesicles are named as telencephalon, diencephalon, mesencephalon, metencephalon, and then myelencephalon. What are the adult derivatives? These are all embryological things. No, in fetal life, in intrauterine life, around 5 weeks, we have the 5 vesicles. So what is these 5 vesicles going to develop in the adult? Okay, these are the adult derivatives. The cerebrum is formed from the telencephalon, whereas the diencephalon is going to form thalamus, hypothalamus, and epithalamus. Epithalamus will not be visible in the sections and all. Okay, but it is going to form thalamus, hypothalamus, and epithalamus. And then from the mesencephalon, we are going to get the midbrain. Then metencephalon, it is going to form pons and cerebellum, and myelencephalon, it is going to form medulla oblongata. Again, these are all will be asked in a MCQs, and then also viva and spotters also can be kept, and then they can ask the adult derivatives for the first year students. For the final year students and all, the derivatives of telencephalon, diencephalon, mesencephalon, metencephalon, myelencephalon is very important. Okay, I'll revise again. Telencephalon, cerebrum is formed. Okay, the cerebral cortex. What we say is the cerebral cortex is formed from telencephalon. And then diencephalon is going to form thalamus, hypothalamus, epithalamus. Mesencephalon is going to form the midbrain, and metencephalon is going to form the pons and cerebellum. Myelencephalon is going to form the medulla oblongata. So this is a beautiful picture from Gray's Anatomy. This is the picture from Gray's Anatomy. Okay, and this is from the internet. I got the source. And this is actually a developing embryo. The 5 weeks embryo is seen in the uterine cavity. Okay, in the intrauterine life. Okay, this is a 5 weeks embryo. At the end of 3rd week, what happens is the embryo will undergo foldings. Okay, there are 2 types of foldings. The embryo will be folding along the cranial and the caudal position. This is called as craniocaudal folding, and then the folding will happen along the lateral ends. Okay, the lateral ends of embryo, it will unite to form the lateral foldings. Okay, the lateral foldings and the craniocaudal folding happens at the end of 3rd week. Okay, and then what happens is the brain. This is the cranial end of the brain, no, which is going to form the cerebral cortex, and then we are going to have the thalamus. We are going to have the midbrain and all. Okay, this is about the cranial end of the neural tube, which also undergoes folding. When the embryo folds, this also will be forming a folding and all. So when it folds, it will be forming the flexures. That is called as cephalic flexure. What you are seeing here is the cephalic flexure. And then here they have opened the forebrain. Okay, the anterior neuropore. Actually, this is the anterior neuropore before the closure. So when the anterior neuropore closes, it will be in the adult, it can be traced as lamina terminalis. So the closure happens at the level of lamina terminalis. The anterior neuropore closes, and this open structure, no, it will become a closed cavity. Okay, so it will then become a closed cavity. Initially, it is an open cavity. So when the closure happens, it will become a closed cavity. Okay. So this is the forebrain vesicles, and then midbrain vesicles, and then this is the hindbrain vesicle. So you can actually also note out all these things. What are these? These are the cranial nerve rootlets. We have cranial nerves and the spinal nerves. We have 12 pairs of cranial nerves. No. So all these cranial nerves will be emerging from the either from the brainstem. Okay, a part of a brainstem, that is from the midbrain, pons, and the medulla. Okay, and then only two nerves, it will be related to the near above the midbrain, it will be related. Okay, so what you are seeing are the rootlets of cranial nerves. So this is third cranial nerve. What is third cranial nerve? Oculomotor nerve. And then you can also see the fourth cranial nerve, that is trochlear. And then fifth cranial nerve. These are all the nucleus. So whenever we are talking about the nervous system, you have to get oriented to like it has an gray matter and then white matter. So what is gray matter? Collection of nerve cell bodies. Okay, only the cell bodies will be collected in the gray matter, whereas the axons, the myelinated axons and all will be collected as a nerve tracts in the white matter. So we have the cranial nerve nucleus at this levels. So these are the rootlets that is just going to form the cranial nerve. Okay, so it is at the developmental stage. So this is fifth cranial nerve, trigeminal. And then we have the seventh and eighth cranial nerves. That is facial is the seventh cranial nerve. Eighth cranial nerve will be vestibulocochlear nerve. And then ninth cranial nerve is glossopharyngeal. Tenth nerve nucleus is forming here. Okay, this is vagus nerve. And then 11th cranial nerve is forming here. This is the spinal accessory nerve nucleus. It is forming here. And then here it is going to form the hypoglossal nerve nucleus. That is the 12th cranial nerve. Okay, this is actually forming developmental stage. And this is again about the development. At around 25 stages, it is only the neural tube. It is like a neural tube. And then 35 days, it is going to form the cranial, the cranial end. It is actually folding here. Okay. The cranial end is folding here. The pharyngeal apparatus, it is forming at the neck level. Okay. And then it is going to form all these flexures and all. And then next few days, that is 40 days and 50 days and all, it is going to form the vesicles. That is the forebrain, midbrain, hindbrain vesicles. And then it is subdivided into telencephalon, diencephalon, metencephalon, myelencephalon, and then mesencephalon. And then 100 days and all these has been formed. What? All the vesicle subdivision has been formed, all the flexures has been formed. And then it is going to undergo a further development. It is going to further increase in size. At around 5 months and all, it will be increasing in size. Okay, around 6 months also, till 7 months, the brain and the skull will be increasing in size in equal orientation. Okay, the skull also, it increases in size. The brain also increases in size. Till then, we need not need a sulci and gyri. Okay, after 7 months, what happens? The brain will be growing. It will be growing. It has to get adapted into the cranial cavity because the skull is actually entirely formed now at around 8 months. After this, the brain has to get adapted into the cranial cavity. It doesn't have space there. So what happens? It will be throwing itself into numerous fold. Okay, the numerous fold formed are called as sulci and gyri. The gyri is nothing but an elevation. The sulci or the depression. At around 9 months, all the sulci and gyri has been formed. Okay, till 7 months, there is no sulci and gyri. Only the brain is increasing in size. What happens is after 8 months and all, the sulci and gyri, it is going to be formed. Okay. Moving on to the anatomy. You all know cerebral cortex. It has two cerebral hemispheres. The cortex, the cerebrum, it is called as telencephalon. That is the end brain. Okay, it has two cerebral hemispheres, which is actually divided into two halves. Two equal halves of cerebral hemispheres are there. Okay, there are two equal halves of cerebral hemispheres. Okay, and then about sectional anatomy, we are going to see the anatomy in different sections. Okay, in different sections. Along the sagittal suture, if the slice is made, okay, if the brain is sliced along the sagittal suture, it is called a sagittal section. And if the slice is made along the coronal section, what is meant by coronal? Coronal means crown. Okay, we'll be placing the crown here, no? So along the coronal section, if the brain is sliced, it is called as coronal section. And then along the longitudinal fissure, we have two equal halves of cerebral hemispheres. So there is a fissure which will be extending in deep. Okay, that is called as medial longitudinal fissure. So along the medial longitudinal fissure, if the slice is made and then if the cerebral hemispheres are made into two halves, it is called as medial section. medial section. Okay. And one more section is the horizontal section. Okay. So we are cutting here. We are putting a transverse section or horizontal section, and then we are reading the cerebral cortex. Okay. So this is about coronal section. So they have made a coronal section here. They have made a slice here. And what you are seeing is the anterior view. From anterior aspect, after making a section, we are seeing the brain. This is the coronal section of brain. So whenever a section of picture is kept, okay, this section is also can be kept in a CT, MRI also. CT images and then MRI images also will have a coronal section pictures and all. You have to apply your knowledge of anatomy in the radiological pictures. Okay, so we'll see what are all the structures here. Okay, as I told you, there is a median longitudinal fissure which will be extending deep into the substance and then separate the two cerebral hemispheres. So in the midline, what you are seeing is the medial longitudinal fissure. Okay, it is called as longitudinal fissure. And then this is the outer gray matter and then inner white matter. In spinal cord, we can see the gray matter in the inner aspect. It is inner gray matter and outer white matter. But here in the brain, the gray matter will be occupying the outer position. Okay, the outer gray matter, and then inside we have the white matter. And then within the substance of white matter, we have masses of gray matter. There is outer gray matter and then white matter. And then within the masses of white matter, we have the gray matter also. Again, the gray matter is present within the white matter substance. Okay, this is about the coronal section. And then the coverings. Coming to the coverings are different from brain and the spinal cord. There are so many differences. One difference here again is the meningeal coverings. What are the meningeal coverings we were discussing? It has a pia matter, arachnoid matter, and the dura matter. The which layer is very different is the dura. The cranial dura is different from the spinal dura. The cranial dura will be different from the spinal dura. How it is different? Actually, to understand this is the horizontal section where you are seeing the brain within the cranial cavity. Okay, this is about the skull. This is the calvaria. Okay, skull or calvaria. Within the skull, you are seeing the cavity that is called as cranial cavity. The cranial cavity, it is divided into anterior cranial fossa, middle cranial fossa, and posterior cranial fossa. And then the brain will be accommodating the cranial cavity. Okay, so we have the pia matter, arachnoid matter, and the dura matter. The cranial dura has two layers. One layer which will be covering the meninges. One layer which will be covering the meninges, that is called as meningeal layer of dura matter. Another layer which will be covering the periosteum of the bone. You have the bone here, no? So the bone has a covering, that is the periosteal layer. So that this dura, what happens is it will be lining the periosteum also. So that is called as periosteal layer of meningeal dura. Okay, cranial dura. Periosteal layer of cranial dura, and then we have the meningeal layer of cranial dura. But what happens is the cranial dura, it will end at the level of foramen magnum. Only the meningeal layer of the dura matter will be continuous with that of the spinal cord. In the brain, there are two layers, periosteal and the meningeal layer. But at the level of foramen magnum, the periosteal layer will be ending. It will blend with the outer periosteum. But what happens? The spinal cord, it has a continuation of the meningeal layer of the dura matter. Okay. So again, coming to the coronal section. So what you are seeing in the middle, okay, what is this? This is nothing but corpus callosum. What is corpus callosum? It's an example for commissural fibers. What is commissural fibers? It is nothing but the white matter fibers. Okay, we have the white matter fibers which will be connecting the lobes of the cerebral hemispheres. Okay, so we have connections. We have a right hemisphere and then left hemisphere, but we need a connection from the right side and the left side. So these connections are made by the white matter fibers. Okay, they are called as white matter projections. Okay, even the cerebral hemisphere has to connect with the thalamus. It has to get connected with the thalamus. From the thalamus also, it should be connected with the spinal cord and all. So these are all connected through the white matter fibers. They are called as projections. Okay, white matter projections. One of the white matter projection is commissural fibers. The largest commissural fiber is corpus callosum. Okay, what is meant by corpus callosum? Actually, it is derived from a Latin word. We are using the term now, but it is derived from a Latin word meaning tough body. Corpus callosum means tough body. So it is a tough body connecting both the cerebral hemispheres. So this is a corpus callosum. And then we have a cavity. Why there is a cavity in the brain? Initially, it is formed from neural tube. No, it's a tubular structure. The tubular structure, it has formed the vesicles. It has formed how many vesicles? 5 vesicles. From the 5 vesicles only, we are getting the cerebrum, cerebellum, all this midbrain and all. Okay, so we had a cavity. In the embryo itself, we had a cavity. That cavity will be in the future, after all the vesicles, it increases in size and then it forms a cerebrum and all. These cavities, they are going to occupy the central positions. Okay, they are going to occupy the cavities within the cerebrum, cavities within the diencephalon and all. Okay, so one cavity, what you are seeing in the telencephalon is the ventricles. This cavity is called as ventricular system. This is the lateral ventricle. What you are seeing is the lateral ventricle. Okay. So, and then in the midline, on either side of the midline, what you are seeing is the caudate nucleus. So whenever I talk about the caudate nucleus or lentiform nucleus, globus pallidus, putamen and all, this is very new for a first year student. If the first year students, they are attending this session, it will be very new for them. All the brain terminologies will be very, very new for them. Okay. But what I say is, you get oriented to all these things. I'll be repeating all these things in my session also. And also you also can go learn and then discuss with your friends. When you go deep into the structures and all, you'll get oriented to all these words. You'll understand all these meanings. Okay. What is caudate? Why it is called as internal capsule? Why it is called as external capsule? Like that you have to understand. When you understand, it's very easy to remember all those things. Okay. Now coming back. So this is corpus callosum. This cavity is the lateral ventricles. And then on either side, we have the caudate nucleus. That is head of the caudate nucleus. And then lateral to the caudate nucleus, you can see the white matter. Okay, this is a white matter projection, which is called as internal capsule. It is present here. So it is called as internal capsule. Then lateral to that, we have the putamen. And then lateral to the putamen, what we have is the mass of white matter. That is called as external capsule. This is internal capsule. This is external capsule. And then again, we have a thin structure of gray matter that is called as claustrum. Okay, and lateral to the claustrum, again, we have a white matter that is called as extreme capsule. So we have white matter in between the gray matter and all. So the gray matter, they are called as caudate nucleus, and then the putamen and the claustrum. The white matter, they are called as, what is this? Internal capsule, and then external capsule, and then we have the extreme capsule. So lateral to the extreme capsule, what you are seeing is the insular cortex. We are seeing about the cerebral cortex, which has thrown into numerous sulci and gyri. Okay, I told you 2/3 of the part of the brain, it is hidden in within the sulci and gyri. Okay, so one hidden part is the insular cortex. So what you are seeing in the section is the insular cortex. And what are all these things? These are all the cranial nerves. You told, we have the cranial nerve will be emerging from the brainstem and all. This is the optic nerve. Okay, this is the optic nerve, second cranial nerve. And this is the oculomotor, trochlear, trigeminal. And then we have the sixth cranial nerve, obtuse, facial, vestibulocochlear, glossopharyngeal. And then we have the vagus nerve and all. Okay, and then the cranial accessory nerves. These are all the cranial nerves oriented here. Okay, so in the coronal section, we can make out the longitudinal fissure, and then outer gray matter, inner white matter, and then we can see the masses of gray matter here. The cavity here is called as lateral horn, and then we can actually find out the corpus callosum, that's the commissural fibers, and this is the caudate nucleus, internal capsule, putamen, external capsule, claustrum, extreme capsule, and then we have the insular cortex. Here, this is the brain stem. These both are the cerebellum. It is called a small brain. Cerebrum, it is called as large brain, whereas here the cerebellum, it is called a small brain. Okay, and this is the brain stem where you can see the pons and medulla oblongata here. Okay, this is about the coronal section, and then I'm moving on to the horizontal section. So this is about horizontal section. When the brain is sliced like this, okay, when the brain is sliced along this section, you can see this view. This is the horizontal section where the brain is actually made a section here. And we are seeing from the superior aspect. From the superior aspect, we are seeing the brain. Again, what is this? Same, that is longitudinal fissure. And what is this cavity? This is lateral ventricle. So lateral ventricle has horns. This is anterior horn of lateral ventricle, which is continuous with that of the third ventricle. Okay, and then posteriorly, we can actually make out the posterior horn of lateral ventricle. Okay, this is lateral ventricle extending like this. Anterior horn and then posterior horn, In the middle, what you are seeing is the third ventricle. What is this cavity? This is third ventricle. Okay. And there is a projection here. Within the ventricle, there is one projection here. What is this? It is called as interthalamic adhesions. Interthalamic. Both the thalamus on either side, it will be connected in the midline. It is called as interthalamic adhesions. So this is interthalamic adhesions. Okay. And then this is thalamus. What you are seeing here is the thalamus. Okay. And then near the lateral ventricles, what you are seeing is the caudate nucleus. Okay. This is the caudate nucleus. This V-shaped limb. Okay. This V-shaped thing of white matter is nothing but the internal capsule. Here you can actually make out very beautifully the globus pallidus and the putamen. Okay, this is the putamen and the globus pallidus. Medial thing will be the globus pallidus. Laterally, you have the putamen. The globus pallidus is again divided into internum and externum. That is called as globus pallidus internum. Another is globus pallidus externum. Okay, thalamus, internal capsule, globus pallidus internum, globus pallidus externum. And then we have the putamen. Then what you are seeing is the white matter, that is the external capsule. The lateral to that, what you are seeing is the claustrum. And then what you are seeing is the extreme capsule. This is extreme capsule. And this is insular cortex. And what is this extreme capsule? What is this claustrum? And what is this? This is external capsule. And what is this? Putamen. This is globus pallidus externum, globus pallidus internum, and then we have the internal capsule, thalamus, interthalamic adhesions, and then this is the third ventricle. Within the lateral ventricles, you can make out some things. This is the choroid plexus. What is this? Choroid plexus. From the choroid plexus only CSF is produced. Okay, the CSF will be produced and then it will be entering into the ventricles and then it will be entering into the from the lateral ventricle, third ventricle, from the third ventricle, it enters into the fourth ventricle. From fourth ventricle, it is communicated to the central canal of spinal cord. Okay. So this is horizontal section. Can make out all these things now. Median fissure, that is longitudinal fissure. What is this? Corpus callosum. Okay, very good. And then this is lateral ventricles. Okay, this is anterior arm of lateral ventricles. I'm thinking that you would have answered these things. Okay, overall listening to this would have answered, and I'm appreciating you. It's very good like that. Okay, and this is anterior arm of lateral ventricle. And what is this? This is thalamus. And what is this? This is posterior arm of lateral ventricles, and we have the choroid plexus here. Okay, what you are seeing is the choroid plexus. And then this one. What is this? Caudate nucleus. And then this is internal capsule. And this is globus pallidus. This one, putamen, external capsule, claustrum. And then we have the extreme capsule and then insular cortex. Okay. Moving on to surfaces of the brain. What are the surfaces of the brain you know? We have the lateral surface. That is called the supralateral surface. And then when we made a cut section, we are having the two hemispheres. And then we can see the medial surface also. So there is a supralateral surface, and then there is a medial surface, and then there is an inferior surface of brain. Okay, there are inferior surfaces, three surfaces: supralateral, inferior, and then medial surface of brain. And then coming to the lateral surface, what is the importance of all this lateral surface? You can actually make out all the sulcus. The important sulcus on the lateral surface is very, very important. Sulcus means depression. Okay, so this is the central sulcus. Everyone would have heard about the central sulcus. How do you identify the central sulcus? This is the frontal pole of cerebral cortex. Poles. This is the frontal pole, and this will be the occipital pole. And then we have the temporal pole. Okay. So when we take a midpoint from the frontal pole and occipital pole, there is a midpoint. From the midpoint, we have to trace behind 5 centimeters behind the midpoint. Midpoint between the frontal pole and occipital pole has to be identified, and then we have to trace behind 5 centimeters behind the central midpoint. Okay, so that you can identify the central sulcus. This is central sulcus. What is the importance of central sulcus? The important is it divides the two lobes of cerebrum. One lobe is, again, we all know there are 4 lobes for cerebrum. Okay, there is a frontal lobe, temporal lobe, and then parietal lobe, and then occipital lobe. What are the 4 lobes? Frontal, parietal, temporal, and then occipital lobe. There it is different from poles. We have a frontal pole, and then occipital pole, and then temporal pole. What we are seeing is the central sulcus. This central sulcus, it will divide the frontal and the temporal lobe. Okay. Coming to the another important sulcus, that is lateral sulcus. Laterally, one sulcus will be extending like here, that is called as lateral or Sylvian fissure, which is going to divide the frontal lobe from the what lobe? Temporal lobe. Okay. Frontal and temporal lobe is be divided from by the Sylvian fissure. What I told you, central sulcus divides the frontal lobe and the parietal lobe, whereas the Sylvian fissure divides the frontal lobe from the temporal lobe. And then we have the occipital lobe posteriorly. We have the posteriorly occipital lobe. It can be divided by the parieto-occipital sulcus. Parieto-occipital sulcus, which will be dividing the parietal lobe from the occipital lobe. Okay. And then this is about the lateral surface. This is about the lateral surface. This is the lateral fissure extending here. Okay. And then whenever we are reading about the supralateral surface, there are three important sulcus. But other than that, we have the numerous sulcus and gyrus. Only the important, every sulcus and gyra is named. Okay, they are called as Brodmann areas. But Brodmann, okay, this is called as Brodmann areas. But we are not going to read all the areas. But at least we should know the important areas. That is the motor area, sensory area, and then important areas in the frontal lobe, temporal lobe, and then parietal lobe and all. We should have a orientation. So this is very vague. You have to listen. No other way, you have to listen. Okay. So what is this? What are these four lobes first? This is frontal. This is parietal. This is temporal. And this will be occipital. Okay. And this one is the central sulcus. What is this? This is the lateral sulcus. Then we have the parieto-occipital sulcus. So we have to draw imaginary lines. Okay. So from the parieto-occipital sulcus, one imaginary line will be extending till the pre-occipital notch. In front of the occipit, we have a notch here. Okay, that is called as pre-occipital notch. So from the parietal sulcus, we are drawing an imaginary line till the pre-occipital notch. And then second line, we are drawing from here. From where? Lateral sulcus. From the lateral sulcus, we are drawing one imaginary line, which will be joining this another imaginary, first imaginary line. This is the first line. The second line will be joining the first line. So that there is distinguished four lobes: frontal, parietal, temporal, and the occipital. Okay. Now we are going to see the sulcus and gyrus in the frontal lobe. So in front of the central sulcus, we have one important sulcus that is called as precentral sulcus. And other sulcuses are superior frontal sulcus. This is superior frontal sulcus. And this is inferior frontal sulcus. So we are going to see the gyri. Okay, gyri. We have seen the depression and all. So we are going to name the elevations. That is gyri, gyrus. Okay. Between precentral sulcus and then central sulcus, there is an elevation. That gyrus is called as precentral gyrus. That is the motor area. Very important thing is the motor area. There is a precentral gyrus, which is situated here. Okay. And then we are going to see the frontal gyruses. There is a two sulcus, superior frontal sulcus and then inferior frontal sulcus. This two sulcus will divide the frontal area into three gyruses. They are named as superior frontal gyrus, middle frontal gyrus, and then inferior frontal gyrus. What is the importance of inferior frontal gyrus? Actually, in the inferior frontal gyrus, we have the lateral sulcus now. From the lateral sulcus, there are rami. There are ramifications from the lateral sulcus. They are named as anterior ascending. One ramus will be ascending, that is anterior ascending, and then one ramus will be horizontal, that is anterior horizontal, and then another is the posterior ramus of lateral sulcus, which is called as posterior ramus. Okay, this will be dividing the inferior frontal gyrus sometime. Okay, this is about the frontal thing. I'll repeat again. Important sulcus: central sulcus, pre-central sulcus, superior frontal sulcus, and then inferior frontal sulcus. Gyrus: pre-central gyrus, and then we have superior frontal gyrus, middle frontal gyrus, and then inferior frontal gyrus. Near the inferior frontal gyrus only, we have the Broca's area also. That is the Broca's area for speech, for language. Okay, we have the Broca's area. Moving on to parietal lobe. Actually, frontal lobe, the importance of frontal lobe function is mainly about the motor area. We have the motor area now. So it will be maintaining all the motor activities of our body. from various parts of the body. That is, that too mainly the opposite side of the body is controlled by the right side. Okay, we have two gyruses. We have the precentral gyrus on the right side also, and then precentral gyrus on the left side also. We are having, if we are a right-handed individual, our left side brain will be dominant. It is called as left dominant hemisphere. If I am a right-handed individual, my left side of the brain is dominant. I have a precentral gyrus, that is motor area on the left side also. There is a motor area on the right side also. So what happens is, my opposite side of the body is controlled by this one. My left motor area controls the right side of the body. My right motor area controls the left side of the body. So it will be controlling the opposite sides of the body. Okay. So mainly the motor activities. Okay. And also about the language. So I'm talking, I'm speaking. Okay. I'm looking at an object. I'm speaking. I could actually orient what I am seeing, and then which is oriented into my frontal field. From the frontal field, I'm getting my speech. Okay, this is very beautifully organized. This is actually in the brain, these beautiful organization and all, it is going on. Okay, when this organization is disturbed, we'll have difficulties. If the Broca's area is disturbed, okay, if there is a cut in the association fibers, what happens is this orientation will be disturbed, and I cannot, even if I see, I cannot talk. I cannot reciprocate. I cannot, even if I am understanding all those things, I cannot reciprocate. Okay, that disturbance and all will be happening. Okay, so these are got importance of motor activity, and then language control and all. It is controlled mainly by the frontal lobe. So coming to the parietal lobe, what is the parietal lobe is going to do? Parietal lobe has a sensory area. Okay, before that we have to know about the sulcus and the gyrus and all. The important sulcus here is posterior to the central sulcus, we have post-central. This is central sulcus. In front, we have pre-central sulcus. In the frontal area, in the parietal area, we have the post-central sulcus. Between central and the post-central sulcus, there is a gyrus. That is the post-central gyrus, nothing but the sensory area. My sensations. If I'm feeling the heat, if I'm feeling the temperature, it's so hot, it's so cold. These sensations are all carried by our sensory motor system, and then they are actually going to relay at the sensory area. The sensations. When I'm touching a hot object, these are all carried from the periphery, and it is reaching my brain. Where it is reaching? It is reaching the sensory area. Okay, here we have the sensory area. And then from the sensory area, it will be having an association to the motor areas also. Okay, so if I'm touching a hot object, what my motor tract will do? It will send impulses through the corticospinal tract, pyramidal tract. And then from the pyramidal tract, I'll be having the reflexes. This stretch reflex. Okay, I'll be removing my hand from the hot object. Okay, from the hot surface, I'll remove my thing. So these are all oriented here. Okay, this is sensory area and the motor area. So important sulcuses. One is post-central sulcus. Another is we have to know about the intraparietal sulcus. Within the parietal lobe, we have the intraparietal sulcus, which will divide the parietal area into superior parietal lobule and then inferior parietal lobule. And then we have two more gyruses, supramarginal gyrus and then angular gyrus. Supramarginal gyrus is near the tip of the lateral sulcus, and the above area is called a supramarginal gyrus. And then we also have the angular gyrus here. Okay. So this is, what is this? Sensory area, post central gyrus. What is this? What is this sulcus named as? This is actually intraparietal sulcus. Okay. So what is the gyrus here? This is superior parietal lobule. And then here below the sulcus, we have the inferior parietal lobule. And near the tip of the lateral sulcus, supramarginal gyrus. And then here again, posterior to that, we have the angular gyrus. Okay. Moving on to occipital lobe. We have finished frontal, parietal. So moving on to the occipital lobe, we had the parieto-occipital sulcus here. Another important sulcus are lunate sulcus, calcarine sulcus, and then lateral occipital sulcus. The pole will be the occipital pole. Okay. Moving on to the temporal. Temporal also, we have two important sulcuses, like frontal. We had superior frontal and inferior frontal sulcus. No, same way in the temporal lobe also, there is a superior temporal sulcus and then inferior temporal sulcus, which is going to divide the temporal lobe into three gyrus: superior temporal gyrus, middle temporal gyrus, inferior temporal gyrus. Same frontal, same like frontal. We had superior frontal, middle frontal, inferior frontal gyrus. We have temporal gyruses also. Superior temporal gyrus, middle temporal gyrus, and then inferior temporal gyrus. Okay. So what is this? We have seen, no, those are from the lateral sulcus, we had ramus and all. The ramus and all will be dividing these areas. Okay, we have the pars opercularis near operculum, and then pars orbitalis near the orbital region, and then we have the pars triangularis between the orbital and the opercularis. Actually, this is a human brain. Okay, this is a human brain. From the fresh cadaver, they have dissected this one. This is the anterior part. Okay, anterior part where you can see the orbit. This is the orbit. Okay, orbital cavity. From the orbital cavity, can you make out one nerve? From the eyeball, what is this nerve? This is optic nerve. And what is this artery? This is ophthalmic artery. This is ophthalmic artery and optic nerve, and then orbit. Okay, and this is the nose. What you are seeing is the nose here. Okay, and from here, this is the cranial cavity. Within the cranial cavity, you can see the cerebral cortex. Okay, the cerebral cortex. Actually, this is the lateral sulcus extending like this, and then you can actually make out the pars opercularis, pars orbitalis, and then pars triangularis. We have to trace this area. This is pars orbitalis, and then this is pars triangularis. And posteriorly near the operculum, it is pars opercularis. Okay. Moving on to medial surface. Again, these are all very boring, but very, we have to listen. We have to listen, then only we can understand. If we miss a certain point, we cannot follow the next slides and all. Okay, so please concentrate. So I'm going to talk about the medial surface of the brain. So when we are making a section along the medial surface and the medial section, and then we are having the two hemispheres. Okay, so this is the occipital pole, and then in front we have the frontal pole, and then this is the medial surface. And what is this? Corpus callosum. Okay, in the medial surface of the brain, you can see the thick body of corpus callosum. This is a thick body. Okay, corpus callosum means Latin it is called as thick body. It is the largest commissural fibers. Okay, so what are the parts of a corpus callosum? When other second year, third year students and all, if they are attending, they can actually answer. Okay, so what are the parts of a corpus callosum? We have a rostrum, beak-like structure. There is a bend here. This is the genu, and then genu or genu. Okay, and then this is body of corpus callosum, and then here we have the splenium of corpus callosum. Rostrum, genu, body, and then splenium. What is this corpus callosum is going to do? If we don't have a corpus callosum, what happens? What happens if there is agenesis of corpus callosum? How this corpus callosum is so important? We are telling it is connecting the different lobes of corpus callosum. It is connecting the different lobes of cerebral hemispheres. So there is a connection. So we can actually understand there is a connection between right side brain and then left side brain. Okay, this what my right side brain is thinking, it has to be communicated to my left side brain also. All these connections are made through the projection, white matter projections. So one of the projection is corpus callosum. So what happens when there is agenesis of corpus callosum? In certain conditions, in certain embryology conditions and all, there is complete absence of corpus callosum. Those persons will have a difficulty in their intellectual memory. Okay, now they are called as intellectual memory. If there is a decreased IQ, they are called as intellectual. What it is called as? Okay, we don't call them as mental retarded. Okay, mentally retarded term is not being used now. They are intellectually deficient. Okay, intellectual disability. They don't have an intellectual disability. Okay, it is called as intellectual disability. So the patient, the children or an adult can have a IQ, decreased IQ. Okay, so they may have an intellectual disability if there is complete absence of corpus callosum. Okay, and then this is again the medial surface, where you are seeing the rostrum, genu, body, and the splenium of corpus callosum. And the important thing is there is a membranous thing here that is called a septum pellucidum. So along, when you are seeing the medial section of the brain, you have to make out certain points I told you, no. So then only you can orient. Okay, this corpus callosum is here. Okay, the genu of corpus callosum is related like this. The rostrum of the corpus callosum is related like this. Posteriorly, from the splenium, we are going to get the fibers like that. They'll be describing in the books and all. Okay, that time you should have orientation. So this is the medial surface where we have seen the corpus callosum. Then we are moving on to the septum pellucidum, and then this is the fornix. And then here, what is this? This is the thalamus. In the center, you have the interthalamic adhesions. So what is lamina terminalis? What is lamina terminalis? When the cranial neuropore closes, it is represented as lamina terminalis. I told you, no, the closure of cranial neuropore in adult, it will be, actually we can trace that thing, that is the lamina terminalis, which will be continuous with that of the rostrum of the corpus callosum. Okay, and then here we have the optic chiasma. Here this is the mammillary body. Then it is continuing with that of the midbrain, and then we have the pons and medulla oblongata here. And then posteriorly, what is this? This is nothing but the pineal body. From the pineal body, we have the colliculus, superior colliculus and then inferior colliculus. These are the projections, posterior projections. And then this is the superior medullary velum, which is extending into the cerebellum. And this is the inferior medullary velum. This is the fourth ventricle. We are not able to see the third ventricle here because the third ventricle lies between the two thalamus. This is the area for third ventricle. Okay. So it communicates with the fourth ventricle. It communicates with the fourth ventricle through the what? How the communication is made? It is through the cerebral aqueduct. Okay. So this is about the medial surface. Again, we should know about same like supralateral surface. The medial surface also has important sulcus at the gyrus and all. The important sulcuses in the medial surface. Okay, you can listen. At least you can remember at least 5 sulcus. 5 sulcus in the medial surface. One important is we have the cingulate sulcus. The cingulate sulcus will be, the cingulate sulcus extends from the frontal area to the parietal lobe. Okay, from the frontal, medial surface of the frontal region to the parietal lobe, one sulcus extends. That is the, what is this? Cingulate sulcus. Another is important is suprasplenial sulcus. We have the suprasplenial sulcus on the medial surface of parietal lobe. Okay, and then we have another important is calcarine sulcus. The calcarine sulcus, it is on the occipital lobe, near the occipital lobe. Okay, and then we have the rhinal sulcus, and then collateral sulcus, and then we have the occipitotemporal sulcus. Like we had the parieto-occipital sulcus, no, on the lateral surface, that will be extending into the medial surface also. From the lateral surface, it extends into the medial surface, the parieto-occipital sulcus. The same way we have the occipitotemporal sulcus. Occipitotemporal sulcus will be here. What are the important sulcus on the medial surface? First is, what is this? Cingulate. And then what is this? It is suprasplenial. Then this one, it is the extension of parieto-occipital sulcus. Then we have the calcarine sulcus. And then we have the collateral sulcus. This is rhinal sulcus. And then this is occipitotemporal. Cingulate, suprasplenial, parieto-occipital, calcarine, and then this is collateral, rhinal, and then occipitotemporal sulcus. These are the important sulcus on the medial surface. So we are going to read about the gyrus. We have an important gyrus, that is medial frontal gyrus. This cingulate sulcus will separate the cingulate gyrus from the medial frontal gyrus. So above we have medial frontal gyrus, and then above the corpus callosum we have the cingulate gyrus. Okay, this is cingulate gyrus. And then we have the precuneus area here, and then cuneus is present in the occipital lobe. And then in the temporal area, we have the lateral occipitotemporal gyrus. And then we have the medial occipitotemporal gyrus. And this is the parahippocampal gyrus. I'll repeat again. This is parahippocampal. Temporal region, medial surface, parahippocampal gyrus. Then we have the medial occipitotemporal gyrus, and then we have the lateral occipitotemporal gyrus. This is lateral occipitotemporal, middle occipitotemporal, parahippocampal. Here we have the cuneus, and then in the parietal region we have the precuneus, Here, this is the medial frontal gyrus, and this is cingulate gyrus. This is about the inferior surface. There is nothing much important in the inferior surface. We have only the olfactory bulbs. Okay, this is the inferior surface of brain. What we are seeing from the inferior aspect. We are seeing the brain from the inferior aspect. That's the inferior view. Here you can make out the olfactory bulb on the either side. And then here, what is this? Is the optic stalk. Optic stalk and then optic chiasma under. Okay, optic stalk, the optic nerve, optic chiasma, and then optic stalk you can make out. And then this is the anterior perforated substances and all. Okay, and these are all the cranial nerve nucleus. Cranial nerves. So again, this is the inferior view. This is actually again from Gray's Anatomy, where you can see the olfactory bulb and then optic nerve tracts and then optic chiasma. And then you can see the perforated substances, mammillary body, pons, medulla oblongata. You can actually see all this cranial nerves emerging from the brainstem. Okay, all the cranial nerves, they emerge from the brainstem. This is again the inferior view of cerebral cortex. We have removed the cerebellum. This is with the cerebellum and the brainstem and the cranial nerve are all intact in this picture. But here we have removed we have cut the midbrain here. We have removed all the cerebellum here. And then after removing the cerebellum, you are seeing only the inferior surface of cerebrum. This is the inferior surface of cerebral cortex. Again, just this is about the longitudinal fissure, where you can able to see the vascular structures and all. What is this? Anterior cerebral artery. Okay, anterior cerebral artery is here. And the longitudinal fissure also lodges the falx cerebri. The longitudinal fissure, it lodges the falx cerebri. Falx cerebri is nothing but the dural fold. I told you there is a dura mater which has to cover the entire brain and then cerebellum and then brainstem and all. So when it comes here, what happens is it will fold. It will form a fold here. Fold and then it will be reflected back. So when the dura mater is folding like this, it is called as falx cerebri. Falx cerebri means it is like that of a sickle-shaped structure. It is like a sickle-shaped structure. It will be lodging here. Okay. So what you are seeing is this circle of Willis. Okay. Both the vertebral arteries, they are uniting to form the basilar artery here. From the basilar artery, we have the middle cerebral arteries. From the middle cerebral artery, we have the communicating arteries, and then we have the anterior cerebral and the anterior communicating arteries here. So this is the area for circle of Willis. Circle of Willis is a very important 5-mark and all for you. Okay, all the vascular supply and all will be asked as an essay, but circle of Willis alone can be asked as an 5-mark even in the final year also. Not only the first year, even in the final year also, they will ask you about the circle of Willis. Why it is very important? Because the perianeurysm, the aneurysm, the abnormal dilatation of the vessels will cause rupture of this one of the arteries, and there will be a bleeding here. So that is called as perianeurysmal rupture. In case of perianeurysmal rupture, what happens is the blood will be in the subarachnoid space. So it will cause a subarachnoid hemorrhage. The patient will be having a thunderclap headache. So when the perianeurysm ruptures, it is a life-threatening condition where the patient will be having sudden, or one young patient will be having a sudden thunderclap headache. Thunderclap na idi vidunda madri vandu pathna thala valikkum appdin solvaanga illaya. And the madri they'll be having a severe headache, thunderclap headache, and then they'll be presenting to the emergency department. Sometimes they'll have a headache and then they'll become unconscious at all. Okay, they'll become unconscious, and then only the relatives they'll be bringing to the emergency department. When he present to the emergency department in unconscious state, we have to suspect a subarachnoid hemorrhage. Okay, so what is the cause for a subarachnoid hemorrhage? It will be an aneurysmal rupture. Where the aneurysm is present, it is one of the perianeurysms. Perianeurysm is most common in the circle of Willis. Okay, this is the chain of reactions. The perianeurysm from the circle of Willis, one of the artery will be ruptured due to aneurysmal rupture. Due to the rupture, he will be having a subarachnoid hemorrhage, blood in the subarachnoid space. Because of that, the patient is having a thunderclap headache. He will become unconscious, and then he will be presenting to the emergency department. It is an emergency condition, subarachnoid hemorrhage. This is about the lobes of the cerebral cortex: frontal, parietal, temporal, and the occipital lobe. And this is the operculum. Coming to the evolutionary aspect, very important. How we have evolved. How our brain has been changed, how over a time our brain has been changed. So initially what happens is we have the allocortex. Our cerebral cortex had only 3 layers. Our cerebral cortex has only 3 layers. That is the allocortex. Allocortex will be divided into archicortex and then paleocortex. Okay. Allocortex is divided into archicortex and then paleocortex. And then we had it has evolved into 4 to 5 layers. That is called as mesocortex. And now neocortex, it is called as new cortex. The neopallium has 6 layers. Why we need 6 layers? Why this evolutionary changes has been taking place? What is all this about? The archicortex. Archicortex is nothing but the hippocampus. Not all of our brain tissues will have all the 6 layers. Only the cerebral cortex, that is neopallium, we have the 6 layers. Okay, but the hippocampus, which is deep inside our brain, we have the hippocampus, that is the archicortex. It has only 3 layers. And then we have certain parts grouped under mesocortex, which will have only 4 to 5 layers. Okay, it's about the gray matter. The triune brain hypothesis. When we are in the dinosaurs period and all, okay, when the humans are seen in the dinosaurs period or that age, okay, I'm talking about those age, okay, that is the reptilian complex. When human has been evolving, okay, when all the reptiles has been forming, that is called as reptilian complex. What is our main function that time? We have to have a survival. We have to survive among the reptilian complex. Along with all the vigorous things and all, we have to survive. Okay, for the nature of survival, we had that reptilian complex. That is, our brain will have a instinctual behavior. We don't have any particular thing. We don't have any grouped things and all. Our brain will be having an instinctual behavior, and then we'll be aggressive. We'll be beating, we'll be killing the animals. We have to survive. Because we have to survive, we have to kill the animals and all. We'll be having aggressive behavior. And then we are dominant. Among the reptiles and all, we should be very dominant. That is called as dominant nature. So this is about the reptilian complex. And then, and over a period, we have moved on to a paleo-mammalian complex. Mammals. What does mean by mammals? We should have a reproductive capacity. We should expand our generation. Okay, the human generation has to be the Homo sapiens has to be. It should be what to say. We have to reproduce and then we have to increase our population. Okay, that is called as mammalian complex. And then we have to feed our species. Not only we are reproducing, we are feeding them and then we are surviving them. Okay, that is the paleo-mammalian complex. That is motivation, emotion. We are having an emotion towards the opposite sex and all. And then we should have a reproductive behavior and then we should have a parental behavior. Even now, our limbic system is concerned with all this parental behavior, reproductive emotion, our sexual behaviors, everything it is concerned with limbic system. Whereas our cerebellum and then brainstem, it will have aggressive behavior. It will have a dominant action. It will have a typical instinctual behaviors. Okay. So then now we are in the neo-mammalian complex. What is neo-mammalian? We are having a new mammalian complex. What is this? We should have an ability for language. We should have abstract thinking. We should have a planning. We should have a perception. Okay, visual perception, auditory perception, the sensual perception. We are perceiving things. We are perceiving things and we are integrating with our brain. Okay, through the integration, we are planning things and then we are speaking. The language is considered as an important evolutionary aspect. Okay, this is about the triune brain hypothesis. Even now, we have all these things. We have the reptilian complex, paleo-mammalian complex, and neo-mammalian complex. But these are all evolved. What I'm telling is here is we have evolved. Okay, from here to here we have evolved. Okay, that is the evolution. So allocortex means it is most primitive. It is the first species with the cerebral cortex. Okay, it has only 3 layers. Mostly our brain in the dentate gyrus, piriform cortex, and the hippocampus and all. It is grouped under allocortex. What is paleocortex? These layers will be having a 4 to 5 layers. That is a cingulate gyrus will have a mesocortex, and then the neocortex. I told you, we have the 6 layers of the neocortex. That is the 6 cellular layers. We have the stellate cells and then pyramidal cells in the neocortex. So first, From what are the 6 cellular layers? We have the molecular layer, external granular layer, external pyramidal layer, and then internal granular layer, internal pyramidal layer, and the multiform layer. Okay, so first is the molecular. This is the molecular layer. And then second is the granular. We have the granule cells mostly here. There is the external granular layer. And then third layer is external pyramidal. We have the pyramidal cells here also. And then we also have the pyramidal cells in the fifth layer. So this is called as external pyramidal layer. And then fourth layer is called as internal granular. We have external granular now. And then this is internal granular. And then we have the internal pyramidal layer. And then sixth is the multiform layer. That is the multiple cell types. This is about, again, the picture from a Gray's Anatomy, where you can see the 6 layers of cerebral cortex, where you can see all the neurons are interconnected with each other, the cellate cells, pyramidal cells, bed cells. These are the histological cells. We can see only under the microanatomy. Okay, only we can see under a microscope, we can see all the cells. This is the pyramidal cell, which is having all the axons and dendrites and all. This is the cell body. Again, this is 6 layers. This is the Golgi stain, this is Nissl stain, and this is Weigand stain. Picture from Gray's Anatomy showing all the 6 layers: molecular, external granular, molecular, external granular, external pyramidal. Nissl stain and all, we can actually make out the pyramidal cells. Can you see the pyramidal cells? Okay, the cell bodies of pyramidal cells can be seen. And here and all the axons and all can be, the Golgi stain can be, all the axons and all can be visualized. And this is the evolution. The amphibian will have only 1 or 2 layers. But the human brains, we have the 6 layers. Okay, the primates and the humans and all, it is having 6 layers of brain. Comparative anatomy, see how big is our human brain. Okay, so the chimpanzee's brain will be like this. And then can you see the mouse brain? Wow, the sections we are seeing under a microscope are all not about a human section. In the histology and all, what normal we are seeing is only the mouse brain and the rat brain. Okay. We'll be comparing our brain, okay, with their sections. We'll catch a rat or mouse, we'll sacrifice, and from the mouse, we'll get all the organs, tissues, and all. We'll do a processing, and then we are making the slides, histology slides. So whatever histology slides you are seeing, they are not human. They are only from a mouse or rat, which is sacrificed in our labs. Okay, so this is a mouse brain, and this is a human brain. How big our brain is. Coming to the white matter. So white matter, they are all projections. We have 3 types of projections. Association fibers will be connecting the same lobe. Okay, this is the right side hemisphere and left side hemisphere. If it is connected, okay, if the same in the left side brain, if the same one side of the brain, it is connected by association fibers. Okay, but whereas opposite side of the brain is connected by the commissural fibers. And then we have the projection fibers, which will be connecting the different parts of the brain. That is, cerebrum is connected with the thalamus and all through the projection fibers. They are the corticofiber system. Okay. Commissural fibers can be divided into anterior commissure, posterior commissure, habenullar commissure, and corpus callosum. The largest commissure, we have seen the corpus callosum. The parts will be rostrum, genu, body, and the splenium. Anterior commissure, it is going to connect the inferior and the middle temporal gyrus. Posterior commissure, it is present in the preoptic nuclei. These are all very, mainly theory, but you have to know about these things. Commissural fibers, there are 4 types. One is anterior, posterior, habenullar commissure, and then corpus callosum is there. But corpus callosum is very important. You have to learn about the corpus callosum. What is habenullar? What is meaning of the habenullar? Habenullar in the Latin, it means rain. Rain means it is just a stalk of pineal gland. Okay, it will be connecting to the pineal gland. Later on, what they did is, the connections between the pineal gland and all, they have named it as habenullar nuclei. And then the commissure, the white matter in relation to the habenullar nuclei, they have named it as habenullar commissure. Okay. It is just a stalk of pineal gland. And then again, this is about corpus callosum. We have the corpus callosum here. This is a picture which is showing the corpus callosum in the midline, which is giving connections between the 2 brains. Can you see the connections? From all parts of the corpus callosum, the fibers will be extending into the cerebral hemispheres. From rostrum and genu, it is called as forceps minor. And then from the body of the corpus callosum, we have the corona radiata, and then it will be connecting with the corona radiata. And then from splenium of the corpus callosum, we have the forceps minor and all. Okay. And also these are all the corpus callosal fibers. And then coming to the association fibers, we have the short association and then long association fibers. The short association fibers are called connects the adjacent gyri, whereas the long association fibers, they are called as superior longitudinal fasciculus, arcuate fasciculus, inferior longitudinal fasciculus, and then singular fasciculus. Okay, so these are all the fibers. So what you are seeing is the fibers. So we have the short association superior longitudinal fibers, and then we have the singular fasciculus syndrome. Superior longitudinal fasciculus. These are all the fibers, association fibers. Coming to the projection fibers. So the projection fibers, I told you, it will be connecting the different parts of the body. Okay, that is different parts of the nervous system. So this is corona radiata is one example for projection fibers. This is the corona radiata, the internal capsule, which is connecting the thalamus and all. Okay, all these are all the projection fibers. So only in the dissected specimens we can see the projection fibers, but there is a advancement now that is called as diffusion tensor tractography. Diffusion tensor tractography can actually follow only the tracts alone. Okay, only the tract alone can be imaged in a diffusion tensor tractography, where you can actually see all these things alone. So this is about functional areas, frontal lobe, and then this is the parietal lobe, and then temporal lobe, and then we have the occipital lobe and all. This is the lateral fissure, we have the central sulcus. So lobes of cortex, occipital lobe. What is the function? What is the importance of function? Occipital lobe, it has a visual unit. Okay, occipital lobe, it has a visual unit. Temporal lobe, it has the auditory cortex. The temporal lobe, it has the primary auditory cortex. Parietal lobe, it has the somatosensory area. We have the sensory area here, and then we have the motor area in the frontal lobe, and then also we have the frontal eye field and all. Okay, so the occipital lobe will be concerned with visual input. Temporal lobe, it is mainly with the auditory cortex. And then the parietal lobe, we have the somatosensory processing. The frontal lobe, we have the motor activity. This is a whole brain model where our left side, okay, our creative thinking and all, it is made by the right side of the brain. Okay, all our imagination, when we are listening to the songs, our feelings, our emotions, okay, what we can express, everything, it is by the creativity is by the right side of the brain. Whereas our left side of the brain, it is mainly for the language, analytical thinking, and then numbers and all. Okay, so this is a picture showing the right side, our right side brain, mainly for the spatial orientation, and then it is also for the ability to draw pictures. It is responsible for imagination, responsible for musicality, emotions and all. Okay, now if I'm taking a class and if you are imagining, if you are dreaming and all, your right side hemisphere is working. Okay, but some persons, they'll be concentrating in our class. Okay, they'll be having analytical thinking, they'll be memorizing the fact and the names and all. When we take the class itself, they'll be taking all the notes and they'll be memorizing all those things. So when for them and all, the left side hemisphere is responsible. So coming to the association functional areas. So area number Brodmann areas. Okay, what is area number 4? Area number 4 is primary motor cortex. Okay, that is the primary motor area. That is the precentral gyrus. We have the area number 4, primary motor area. And then sensory area means area number 312. Okay. Area number 312 is primary sensory area, somatosensory cortex. And then we have the area number 17. 17 is for visual cortex. Area number 17 in the occipital lobe is for visual cortex. And then area number 41 and 42, it is present in the temporal lobe. That is the superior temporal gyrus. The location, it is in the superior temporal gyrus. That is the primary auditory cortex. I'll repeat. Motor area, the frontal lobe, that is the precentral gyrus. Motor area, area number 4. Area number 312, it is primary sensory area present in the postcentral gyrus. And then we have the area number 17, the occipital lobe for primary visual cortex. And then we have area number 41 and 42 in the superior temporal gyrus. That is temporal lobe. That is mainly for the primary auditory cortex. This is again the central sulcus. In front, we have the motor area. And then at the back, we have the somatosensory area. Area number 312. So this is for voluntary motor control. Area number 4, somatosensory 312. And then we need to know about the location and all. I told you the representation will be, it controls the opposite side of the body, and then it also controls the inverted voluntary movements. Okay. This is the motor homunculus, where you can have the representation and all. So when this part of the precentral gyrus alone, it is sectioned, and then when you are seeing here, you can see the representations of our leg, hand, and then face and all over here. Okay, along the medial side, along the superior aspect, along the lateral aspect, there are some representations. This is called as motor homunculus. In the brain, we have a major representation for face and also for the hands. Okay. Coming to the lesions. What happens when there is a motor area lesion? There will be paralysis, because the corticospinal tract, it will be connected to the corticospinal tract. The corticospinal tract has to come down, and then it only will be producing all the reflexes of the opposite side of the body. If the area, there is a central lesion, when our brain is damaged and there is a lesion to the motor area, all the muscles will be paralyzed. There will be inhibition of the stretch reflex. There will be Babinski's signs and all positive. Okay. And then coming to the area number 6, that is premotor area. It is similar to area number, area number 4, it has a body representations. It mainly plans the voluntary movements. Okay, if I have to do this movement voluntarily, I have to have a planning. That planning will be done by the premotor area. In front of the motor cortex, I have a premotor area that will be planning all my movements at all. If the lesion happens, what happens? There will be again weakness, grasp reflex, Babinski's sign, all this will be positive. Okay. Coming to the supplementary motor area, there is a supplementary motor area, mainly it plans and programs and keeps the motor area memory for the motor act. So when a motor area has to act, it has to have a planning and programming happening in the supplementary motor area. So we have motor area, premotor area, and then supplementary motor area. Okay. So the planning and programming will be taken place in the supplementary motor area, but the action will be done by the motor area. Okay. So even when the supplementary motor area is affected, we don't have a planning, again there will be weakness. If the main motor action is affected, again there will be paralysis, there will be weakness. So this is Broca's area. So the Broca's area is present in the inferior frontal gyrus. Imagine if a patient with the Broca's aphasia, okay, if a patient is unable to say, I am hungry, I am in pain, thank you, or I love you. Okay, I'm being trapped inside my body. Your body does not respond to my command. Okay, I'm having that feeling. I'm hungry. I need to tell someone that I'm hungry and then should have a food. I'm in pain. I'm having a pain. I should tell them. Okay, if I'm not able to tell them, it is called as aphasia. Okay, so this happens when the Broca's area is affected. When the Broca's area is affected, the language communication will be affected, and the patient will not be able to communicate what he is feeling. He can recognize all those things, but he cannot reciprocate. That is Broca's aphasia. Okay, that is area number 44 will be affected. And then coming to the frontal eye field, head rotation, and then hand skill and all, it is concerned with area number 8. Okay, and then this is the prefrontal. I want to tell about the important story about a Phineas Cage. This is about a man who is 25 years old. Okay, he met with an accident in around 1848. This accident took place in 1848. It is very interesting how this accident has takes place. This man was working in a war field. Okay, he is working in a war field and he is dumping all the bomb things, bomb powder. He is dumping the bomb powder with the iron rod into a hole. He is dumping the bomb powder with the iron rod into a hole. Okay, suddenly what happened? It exploded. It detonated. Okay, the bomb powder has exploded. Then, because the bomb powder has exploded, it has reflected back. Okay, the iron rod, now what happens is it has pierced this man's cheek, and then it has pierced his frontal lobe, then it has pierced his skull, and then it has went away, and then it has fell around 80 meters away. The iron rod, because of the detonation, it has pierced his cheek, frontal lobe, brain, and skull, and then it went back. Okay, it has gone and fell down around 80 meters away. But after even, this is the iron rod. This is the iron rod which has pierced his cheek, frontal lobe, brain, and the. But after this, what happened? This man is able to stand up, walk, and then even talk. He can go to the emergency department and then he can actually explain the incident that I has about the incident and all. He would be able to explain. Okay. So then what happened? The doctor was following this case. This is a case study. This was presented as a case study. And then they were following this patient. After the accident, after the recovery, what happened is there is a change in his behavior. That is, initially when he was working under a war field, he was considered as hard-working patient. He was very, what to say, he's very kind. He's very not rude and all. He'll be conserved to speak and all. Okay, he'll have all those activities. He's a very soft person. But after the accident, what happens? He has become a very aggressive person. He has become very rude. He has become a chronic alcoholic. He's not able to go to work and all. He was a hardworking, soft-spoken person. But now he has become an aggressive person. He is not able to concentrate on his works. He has become an alcoholic and all. So there is a behavioral changes. So early in 1840s and all, there is no such researches. After this accident, it has actually, the researchers were taking this case as a thing, and then they were doing research and all. They were not able to recognize where this iron rod has pierced, which lobe it has affected, because we don't have any modalities at that time. So we are not able to trace all those things. Only with the behavioral changes, we are following the patient. Okay, only with the symptoms, we are following the patient. But after 100 years, after the revolution of radiology and that happened, what they did is they have reconstructed the skull. They have done a reconstruction in the skull in 1992. The accident happened in 1848. Okay, the patient died in 1862. At the age of 37, the patient died due to epilepsy, seizures and all. Not because of this accident he died. Because of the alcohol, seizures and all, he has died. He has not taken his medicines. So he has lost follow-up. So he died in the young age. But in 1992, with the evolution of radiology, they have reconstructed this, and then they have identified the lesion happened both the frontal lobes. Both the right and the left lobe has been injured by this thing. They have finalized. So when they finalized, so this was considered as an evolution in neurology researches and all. Okay. So till then, they were thinking only the motor activity, only the planning activity, only the all these the visual connections and the language are only controlled by the frontal lobe. But after this only, they have come to a conclusion that the frontal lobe is involved in behavioral changes also. Even now, in neurosurgery departments, when you are posted, you can see the patients. In case of accidental injury and all, if they're having a lesion in the frontal lobe, they'll be behaving very rude, very aggressive. They'll have increased sexual arousal and all. They'll be running here and there. They'll be pulling all the rails and all. Okay, they'll not be them. After their recovery, they'll be completely a different person, because the frontal lobe is now considered to control all the behavioral activities also. This Phineas Cage accident is considered as a revolution in case of neurology. So that I wanted to tell you now. And then coming to the one story. What is this picture about? This is from a famous film, Notebook. Okay, the Notebook story, it is considered as, it is a love story where this lady, she will forget everything due to Alzheimer's disease. Due to aging and all, she'll forget everything. In her infancy, that is during her adulthood and all, she's a brilliant child. And then she was a brilliant child and she was in love with this person. Okay. What happens is, over the time, this person will go to a war and they had a love breakup. And after a period of time, when he comes back, she will ask, oh, who are you? Like this. Okay. She'll completely forget this person. And then they'll be reading this notebook story. From this notebook story, they'll be regaining all their love things and all. And then at the end, they'll join. Okay. This is about the story. Why I want to tell here it is about the Alzheimer's disease. Not only the accidental injury can cause a lesion to the frontal lobe. Okay. Even the degenerative diseases, that is aging also, can cause Alzheimer's, and we can lose our memory, thinking capacities, and then we can lose our language. We can have a change in our behavioral changes and all. Okay. So the prefrontal lesions and all, inability to perform two works at a time, inability to follow the proper sequence of achieving a task. There is lack of initiative. The patient will be very sedentary. He will have a lack of initiative. There is impairment of moral sense. They don't know what they are doing. There is impairment of social sense. They don't know at this place I should not do this. Like in ICU and all, they'll be urinating. Okay, they are admitted in ICU. The frontal lobe lesion person and all, what they'll do is they'll urinate in a public place. They'll urinate in front of a staff nurse. Okay, they will not have a social sense. They will not have a moral sense and all. They will lose everything. So the failure to realize the gravity of situation, they'll have a decreased smell and all. Okay, these are all things due to prefrontal lesion. And then they cannot have a memory storage also, fail to track even bits of information also they cannot track. Even what is your name, what did you had in the morning, all those things and all they cannot have in mind. They cannot reply to you. They'll be easily distracted. Okay. And then inability to plan for future and then all those things. So sensory area, it is again having the visual area, auditory area, vestibular area, gustatory area, and the olfactory. And then for the smell also it has areas. Okay. These are the sensory areas. These are the special senses. The general sensations, it will reach the somatosensory area. That is area number 312. But visual is special sensation. Auditory, hearing is again special sensation. Equilibrium, balance is a special sensation. Taste is a special sensation. Gustatory, and then olfaction, smell. All these areas are in the sensory. These are all considered as the sensory areas. So the sensory cortex, it is mainly at the postcentral gyrus, I told you, and then it will have mainly the general sensations includes tactile localization, discrimination, proprioception, vibration, stereognosis, pressure, muscle tension, pain, and then temperature. All these are sensations, general sensations. So again, they have a like motor homunculus, sensory homunculus is also present as the same thing. These are all about, then we have the association areas also in the parietal lobe. Okay. And then the important thing is Wernicke's area. Okay. The Wernicke's area is language comprehension and then intelligence. All these are concerned with Wernicke's area. Okay. So this is apraxia. Apraxia is inability to execute a voluntary motor movement, being able to demonstrate normal muscle function. I will be having the normal muscle function, but I will not able to execute a voluntary motor function. Like I want to have wear a shirt. Okay. I will be, my motor movements are normal, but because there is there is a problem in the frontal lobe and the temporal lobe, I cannot consider. Okay, I cannot take that and then I can actually wear my buttons. That is called as apraxia. That is inability to execute a voluntary motor movement, despite being able to have a normal muscle function at all. Okay, and then occipital lobe, it is mainly concerned with visual area, and then temporal lobe, it is mainly concerned with auditory cortex, and then speech. Okay, that's all. Thank you.
Ma'am. Yes, ma'am. The way of your teaching, the presentation, and the pictures on the presentation, ma'am, it was really mesmerizing, and I can see the amount of efforts you put in this slide, ma'am. And as you said, the circular fill is very important question. I can still remember it was asked in my university examination. It was a very good session, ma'am. Thank you so much. For the past 1 hour, you have enlightened us, ma'am. And இது மாதிரி pictures நான் வேற எந்த இதுலயுமே பார்த்தது இல்ல. It was really nice. And I thank all the students and doctors those who attended this session. And I thank our organizers, Dr. Abulasan sir, Dr. Venkatesh Karthikeyan sir on this occasion. And thank you everyone. We will meet with another topic. Thank you.
Okay, thank you so much. Can we end the session?
Yes, ma'am.
Okay, thank you.
Thank you.